Hi, how's everyone? Let's have a complete walk through the January 2026 paper of Unit 3 Practical Skills in Biology. For the first question, the blue lotus is a freshwater plant found in lakes and rivers in East Africa, and he showed us a photograph showing its fruits and seeds. The fruit has been used in traditional medicine, which means that we've taken this fruit, crushed and grind it, and dissolved it in a specific solvent in order to get the extract used in traditional medicine. The antibacterial properties of an extract of the seeds were investigated. The extract was made using ethanol. So ethanol now is our solvent that's required for this process. The flowchart shows the steps taken. Bacteria were added to a petri dish containing nutrient agar to make alone. An extract of some seeds was made using ethanol, and we used filter paper discs to be soaked in different concentrations of the extract or a known antibiotic. So now I know that the independent variable is using different concentrations. And I'm comparing it also to a known antibiotic at the end. The discs were left for the ethanol to evaporate. The discs were placed on the bacterial lawn, and the dishes were incubated at 37 degrees Celsius for 24 hours. The diameters of the clear zones, which are the zones where no bacteria grow in anymore that developed, were recorded. First question is describe how to make a clear solution of known concentrations. So take care of these slight details in the question. It needs us to get a known concentration. So what is the concentration? Concentration is a specific mass over a specific volume. So if I need the concentration to be known and controlled, I need to control two things, the mass and the volume. So in the question, I'm firstly going to mention how we're going to crush the seed. So powder the seeds with mortar and pestle. These are the mortar and pestle. Then use a known mass of the powdered seeds and a known volume of ethanol. Take care that we used ethanol here. Water was rejected in the marking scheme, and this is why the question clearly clarified that we are using ethanol. Okay? The product is then filtered using filter paper. Normally, we filtered the extract we got. Describe how bacteria could have been added to the nutrient agar to produce alone. So if I'm going to add the bacteria, I need a suitable tool. My tool is going to be the inoculating loop. And since we're talking about bacteria, so there is a specific idea I need to mention, which is the aseptic technique. I have to mention at least one thing about the aseptic technique, whether to sterilize, use the Bunsen burner. And now this is how I described it. Use a sterile tool, the sterile inoculating loop. And by the way, this is the inoculating loop. I use it here to get the bacteria from the culture. Use Bunsen burner, which is the device we use to get the blue flame, or like any kind of flame, but the blue flame is the strongest, to sterilize the inoculating loop. Then I'm going to use a glass spreader to make a lawn and distribute the bacteria evenly by streaking method. This wasn't mentioned in the marking scheme, but I think we can add it. This is the glass spreader that I'm going to use, and I'm going to use the streaking method. Streaking method is that I should distribute the bacteria in that way in order to be evenly distributed along the nutrient petri dish. Alright, next question. Explain why an incubation temperature of 37 degrees was used in this investigation. So 37 degrees is of course the number of the body temperature. The body temperature is always 37 degrees normally. So 37 degrees would be optimum for the enzymes in this bacteria. Since these bacteria are types of infectious pathogenic bacteria, so this must be why they live in the temperature that's suitable for them inside the human body, for example. Okay, the diagram shows results for two types of pathogenic bacteria, S. typhi, which causes typhoid fever, and V. cholerae, which causes cholera. And we can see that we have the filter paper discs distributed, and these are the zones of inhibition seen. Let's see the questions. Complete the table to show the results for disc 1 and disc 2. Okay, for disc 1, I chose number 11. It's going to be very relative here. That I can see that the disc 1 for S-typhy here is somehow less zone inhibition than number 1 in V. cauleri, and at the same time it's also bigger than number 2 here. It looks bigger than number 2 and less than number 1 in V. cauleri. So I choose a number in between them, which is 11. And for number 2, which looks relatively less than number 2 here, so I'm going to choose a less number than 2 in V. cauleri. What is a smaller number? It would be 10. Suggest how disc 6 was prepared for use in this investigation. Disc 6 has 0 concentration of extract, which means this was used as a control. So how am I going to prepare a control here? It's going to only be the solvent alone with nothing added. So what is my solvent? My solvent was ethanol. So just say that we're going to add ethanol to this disc to act as a control. Okay, next question. Plot a suitable graph, and this is the plotted graph, normally simply drawn. Just make sure you don't forget to add the scale in correct numbers. The labels for axes should be added with their units, and make sure to have accurate plotting of the points. I prefer using X or this one to be more accurate. Then join your points, and don't forget to join using a straight ruler. Starting from 0 point, since we used a control. This is not an extrapolation, by the way. It is not an extrapolation. It is just because we used number 0 here for disc number 6. Containing 0 concentration of the extract. Describe 3 conclusions about the effectiveness of this extract and use the results in the table. Make sure to use the guide they give you in one sentence below the main question. It asks us to use the results in the table, so that's what I actually should use. Okay, the table, I'm gonna get back to the table. And I can see that V. cauleri is generally more affected, while the S-typhy is only affected for these 2 concentrations. Using lesser concentration of the extract is not going to help at all for the S-typhy, but for the V. cauleri it's still working. So I should add this as a conclusion, that V. cauleri is more affected by this extract than S-typhy. Also, you could say that as the concentration of the extract increases, the effectiveness increases, and we don't forget to compare it with the antibiotic we used. The known antibiotic shows a significantly higher effect and higher diameter of the zone of inhibition. So that's also another point I should add, that antibiotic is more effective than the extract. Now we're done with the first question. Let's keep going. A study found that many adults consumed an unhealthy takeaway meal every day. The photograph shows a typical takeaway meal consisting of a burger with cheese and a bread bun, fries, and sugar-free soft drink. A student decided to investigate the carbohydrate, protein, and fat composition of a takeaway meal. The meal was liquidized in a blender, the mixture produced was filtered, and the filters was tested for the presence of carbohydrates, proteins, and fats. First question is pretty direct. Complete the table to show a suitable semi-quantitative test for each food type. Fats are going to be tested using the emulsion test, and the other tests are memorized. Reducing sugars is using the Benedict's test, Benedict's reagent used, which shows a range of colors. If you remember, highest is brick red. Starch is tested using iodine test, and protein is tested using the Biuret test. That's it for this question. Pretty direct and simple. You should get the marks easily. A protein test was performed on a sample of the filtrate, and the result compared with color standards. The diagram shows the results for the filtrate and standard solutions of proteins. or estimate the concentration of protein in the filtrate. This is my filtrate, and I can see that the color is intermediate between 40 and 50. So I can just choose any number in between. It could be from 41 to 49. I chose 45 milligram centimeter cube here. The total volume of the filtrate was 350 centimeter cube. Calculate the mass of protein in the filtrate, and don't forget to give your answer in grams. This is quite the trick here that makes all of you lose 1 mark for no absolute reason. This is pretty simple as a math equation where concentration equals mass over volume. Just substitute into this equation. I have the concentration, which I estimated here, and I have also the volume of the filtrate, which is 350. If I want to get the mass, I'm just going to multiply 350 times 45, but take care that this is not the final answer, since the volume I had was given. Oh yeah, the problem is not in the volume, that I have to convert it from milligram to grams. So the conversion from milligram to gram is when you divide by 1000. So make sure to divide by 1000 to get the final answer, which is 15.75. If you want to round it up to 3 significant figures, you're gonna get 15.8, and this is going to be my final answer. Another pretty direct question, and it was actually repeated in many past papers I've seen before. Describe the difference between a quantitative and a semi-quantitative test. We're just going to give a brief description of each. A semi-quantitative test gives an estimate of the concentration, while a quantitative test, which is usually given from a colorimeter, gives the exact value of concentration. My main key points here are estimate and exact. So the semi-quantitative is an estimate and the quantitative is the exact value. Alright, let's continue. The concentration of reducing sugar in the filtrate in gram per decimeter cube was found. The table shows the appearance of standard solutions of reducing sugar when tested. I have the appearance of all of them linked to each concentration of reducing sugar. The question here says the mass of reducing sugar is in the 350 centimeter cube of filtrate was 5.6. State the appearance. So this means that I have to get back to the table, but the table doesn't have the numbers in the mass or in the volume. It has it in the form of concentration. So that means I'm gonna have to calculate the concentration in order to find the appearance of this filtrate. So I'm gonna divide mass over volume again, but take care here that the units are in gram per decimeter cube, so you should also convert from centimeter cube to decimeter cube, and this is done by dividing by 1000. So if I divide the 350 centimeters cube by 1000, I get the 0.35. Concentration is here mass over volume, gives me 16 gram per decimeter cube. I can now accurately get it, and it's now orange with precipitate, so just write this. The filtrate was found to contain 36 grams of carbohydrates, 5.6 of them was reducing sugar. And this is quite a silly question. I couldn't understand the idea of the person giving this question actually. Give one reason why the mass of reducing sugar is different from the mass of the carbohydrate. The answer is also pretty simple. The carbohydrate content may include non-reducing sugars as well. You can give examples if you want, but it wasn't awarded any marks in the marking scheme, and they could be sucrose, starch, or cellulose. Resistant starch is a form of starch that is difficult to break. Resistant starch is found in potatoes. In an investigation, the effect of the variety of potato, the cooking method used, temperature after cooking, on the mass of resistant starch was determined. The table on the next page shows the results of this investigation. I can see that I have lots of standard deviation values, and this gives me a hint that I'm going to use them. For now, justify two conclusions that can be made from the results shown in the table. I can actually get not only 2 conclusions, I can get 3. But for now, we have 2 conclusions, so that's what I'm gonna do. My conclusions are gonna be based on the variety, cooking method, temperature, if I wanna add it, but we only have space for 2, so let's conclude for each one and give the justification. Justification, by the way, refers to using the standard deviation and overlapping idea. And this is how I justify the differences between them. My conclusion 1 was that there is no significant difference between the 3 varieties. I can't see a significant difference between the varieties. These are the varieties, so if I want to compare between each one of them for the hot temperature, for example, I can see that all of them are very similar. That's 3.8, and there's a range of 0.6 higher and lesser. I have 3.5 and 3.5, so that's very obvious. I don't even have to calculate the values after increase and decrease of the standard deviation. So I can see that the standard deviations overlap in all cases. That's why there is no significant difference between them. So using each variety of them doesn't really matter in my results. My second conclusion is that baking leads to significantly more resistant starch than frying. And this is also going to be based on the results that I can see for baking here. The values are higher, and if I want to compare between each one of them, let's say we're going to compare between the cold temperatures for baked and fried, you're gonna find out that if I add 4.8, if I subtract 4.8 minus 0.7, it's gonna be 4.1. And if I want to get the higher value of this one to see if they overlap, that's 3.1 plus 0.2, so that's going to be 3.3. There is no overlapping between the minimum point of the higher number and the maximum point of the lower number. So there is no overlapping, and that means there's significantly more resistant starch than frying for baking. Okay, and this is because the standard deviations don't overlap in all cases. And this is also a very direct question from Unit 1. Describe how a triglyceride molecule is made from its component molecules. And I'm just gonna briefly describe how it's formed. I need 3 from the free fatty acids, and I only need 1 glycerol to form the triglyceride molecule by condensation reaction, and there are ester bonds going to be formed. Give one difference between a saturated and an unsaturated fat. We can simply say that the saturated have no double bonds. Here is an illustrative picture. If you forgot, the unsaturated has at least 1 double bond, while the saturated doesn't have any double bonds. Okay, let's continue. Savinase is an enzyme used in biological washing powders. This enzyme digests protein-based stains on clothing. An extract from an information sheet about Savinase said, it is effective under most alkaline conditions and is very effective at medium temperatures. Its optimal activity is at 55 degrees Celsius, and it can also produce blisters on the skin. This gives me a hint of the safety I'm going to use. If I have any safety issues, it's going to be that I have to wear goggles. No, not really goggles, but we have to wear gloves to avoid the blister effect of the Savinase. And also I have a hint of which pH it prefers, which pH is the Savinase optimum at, and its optimal activity as well. The progress of protein digestion can be followed using a calorimeter, and this is a quantitative test to measure the cloudiness of a protein solution. As the insoluble protein is digested, the cloudiness measured as absorbance decreases. Absorbance is 0 when all the protein has been digested. This is a calorimeter. Explain why the digestion of protein by Savinase produces a clear solution. Why do I have a clear solution? Well, the protein gives a cloudy appearance, and when it's digested, the cloudiness disappears. This is because the protein is, of course, digested into amino acids that are soluble, so that they don't give a cloudy appearance in the solution. The protein is broken down into amino acids which are soluble. Okay, simple question as well. The graph shows the effect of substrate concentration on the initial rate of reaction with Savinase. And this is a graph. It's going to ask us to draw a table, pretty simple. Just make sure you don't forget the units here and make sure that your values are accurate, extracted from the table. Okay, explain why the initial rate of reaction was measured in this investigation. In every question that asks us why the initial rate was measured, it's, of course, firstly, to ensure that the results are valid. And why is that? If you still don't understand this point, even though it was repeated in many past papers, let's explain it. Firstly, when I keep increasing substrate concentration, of course, the rate of reaction keeps increasing at first. The reaction velocity increase is linear at low substrate concentration, and when I keep increasing, yeah, it's okay, it's increasing. But at very high substrate concentration, any additional increase in the substrate will not increase the reaction velocity. And why will it not increase? Because enzymes' active sites are now limiting. I don't have enough active sites for all these substrates to bind. They are all occupied. So this is the idea. I don't want this to happen. So I only want the initial rate to be measured, and this is the most valid part where there are no limiting factors affecting the rate of reaction. As the reaction proceeds, there are also some end products. You should also know this point, that the end products that are produced from this reaction limit the activity of the enzyme. When the enzyme function is starting to work, it's starting to produce some end products. This makes the end products a feedback inhibition for the enzyme. This is also an idea you can take into account, and it was written in the marking scheme actually. So I could say this is to ensure the results are valid because substrate is in excess initially, but as the reaction proceeds, it becomes limiting to reaction rate. Also, there are no end products to inhibit the enzyme yet, so that's what makes the reaction valid at first. Explain the relationship shown in the graph. And let's get back to how we make command words really important. Command words like explain doesn't only need me to explain. In Edexcel, it needs me to describe first. I have to describe the relationship, then I have to explain what I've described. Okay, so it's two things in one. Firstly, I'm going to describe, my description is in blue and my explanation is in pink. Initially, the rate of reaction increases as the reaction proceeds. I can see that it's increasing steeply also. What makes the rate of reaction increase steeply? This is because there are more enzyme substrate complexes, of course. There is more substrate molecules available to collide with the enzyme. But eventually, at last, I can see that it is no longer increasing. It has leveled off. Why did it level off? It leveled off because all the enzyme active sites are occupied now, so we cannot form any more enzyme substrate complexes. And this gets us back to the idea we've described here. Last question. Devise a procedure to show that the optimum temperature for Savinase is 55 degrees Celsius. You are provided with the Savinase solution of the enzyme in a known concentration and the known concentration of the protein solution as well. So that's also kind of an important idea, guys. I don't need to mention this as a controlled variable because it was already controlled. So how are you going to control it when you are already given a known concentration? Okay, so I have to look for another point to control. A colorimeter, which is the device and standard laboratory equipment and chemicals. We have chemicals, so of course we can say that I have buffer, for example, to control the pH. Firstly, use a range of at least 5 temperatures, and I usually standardize this number of 5 in most experiments. Just use 5 and you can give them the range of values you're going to use: 40, 45, 50, 55, 60. That would be great. Before mixing the protein and enzyme solutions together, we have to equilibrate them. Make sure that both the protein and the enzyme are having the same temperature, okay, in order for the reaction to be valid and the results to be accurate. To the temperature using a water bath before mixing. This is also one of the equipment we're given from the standard laboratory equipment and chemicals. Control pH of the solution using pH buffer. I could also say I have to make it alkaline, and this was mentioned in the question that the pH its most great at, its most optimum was alkaline. Okay, so I can make it optimum for its optimal pH for the temperature to be the only effect affecting the experiment. Mix the solutions and use a colorimeter to monitor the absorbance against time. Now I have absorbance against time, and this is going to be the graph I'm going to plot in order to see my results. And don't forget, the results are for the initial rate of reaction. Obtain initial rate of reaction from the graph using a tangent to get the gradient. Also, don't forget the safety issues we have described. Make sure to wear gloves, and this is to avoid the blisters we have seen in the poster, and safety goggles to protect from chemicals splashing. This is how the colorimeter looks like, and this is the whole idea of the colorimetry and how we get the result as a numerical number. After that, we're going to plot the numerical numbers we have seen. It looks like that. We have only one test here, so one graph. It's time against absorbance, or absorbance over time actually. Okay guys, that's it for this video. I hope you found it useful. Thank you guys for watching.